The overall goal of this procedure is to measure the kinetics of mRNA nuclear export in mammalian cells. This is achieved by first microinjecting mRNA or DNA into the nuclei of mammalian tissue culture cells. Following microinjection, the samples are fixed after various incubation periods. The mRNA is then stained by fluorescent in situ hybridization, or FISH. The final step of the procedure is to image the cells and quantify the amount of fluorescence in the cytoplasm and nucleus. Ultimately, the rate of mRNA export can be measured through a combination of microinjection and fluorescent microscopy.
Hi, I'm Serge Garasov from the laboratory of Alex Palazzo in the Department of Biochemistry at the University of Toronto.
I'm Stefan Tarnowski, also from the Palazzo lab.
I am Amy Cui from the Palazzo lab.
And I'm Kohila Mahadevan from the Palazzo lab.
Today we will show you a procedure for introducing mRNA into the nuclei of mammalian tissue culture cells by microinjection.
We will also demonstrate how to stain the mRNA by fluorescent in situ hybridization.
We use these procedures in our laboratory to study the kinetics of mRNA nuclear export in mammalian cells.
So, let's get started.
Prior to microinjection, dilute previously prepared plasmid DNA to between 50 and 200 micrograms per milliliter in injection buffer. Next, add Oregon Green 488, or OG, conjugated to 70 kilodalton dextran to the mixture to enable the identification of injected cells. The OG conjugated dextran will help determine how much of the fluid was injected into the nucleus and how much leaked into the cytoplasm. Centrifuge the samples at 13,000 G and 4 degrees Celsius for at least 20 minutes to pellet any particulate matter that may potentially obstruct the needle tip. Choose the mammalian cell line to be microinjected based on the criteria described in the written protocol. COS7 cells are used for this demonstration. Seed the cells on acid-washed cover slips in 30 mm Petri dishes at least 24 hours prior to microinjection. Ideally, the cell monolayer should be roughly 70 to 90% confluent at the time of injection. To allow for easy identification of the injected cells, the cell monolayer is wounded prior to injection by using a 200 microliter plastic pipette tip. Etch a cross wound on the cover slip and leave the cells to recover in the tissue culture incubator for at least 15 minutes prior to injection. For microinjection, use an inverted microscope that is isolated on an air table to minimize vibrations that can be disruptive to the microinjection process. Using an Eppendorf gel loader tip, remove approximately 1 microliter from the top of the centrifuged sample to avoid disrupting the pellet. Insert the tip of the pipette into the back end of a prefabricated microinjection needle and eject the liquid. The liquid will be drawn into the needle tip by capillary action. A meniscus near the tip should be visible within 5 to 30 seconds. Place the fluid-filled needle into the wand and clamp it to the micromanipulator at a 45-degree angle. Visualize the needle using the 10x objective and position it in the center of the viewing field near the focal plane using the coarse micromanipulator knobs. The wand is connected by a tube to a glass syringe, which is used to generate the pressure that is required to eject the fluid from the needle tip. To maintain a steady pressure, the syringe is mounted onto a pipe clamp using 2 rubber stoppers and 2 metal clamps. Raise the needle a few millimeters to prevent it from being damaged in later steps, and then push back the microscope back pillar. Increase the pressure by depressing the syringe plunger 0.5 to 2 cubic centimeters. Place a petri dish containing a cover slip with a wounded monolayer on the viewing stage. Pull the microscope back pillar forward to an upright position, causing the condenser to be aligned and the needle to enter the liquid. This should be done carefully to prevent the needle from breaking on the cover slip. Using the 10x objective, identify the center of the cross wound and position the needle above the cells to be injected. Increase the magnification by switching to the 40x objective. Then, lower and center the needle using the fine adjustment knobs on the joystick. Begin injection at one corner of the wound cross by lowering the needle to make contact with the nucleus. Once injected, a cell has a notable change in phase brightness. Continue along one edge of the wound cross for easy identification of injected cells during visualization. It is critical that the injection pressure be properly adjusted for each needle. The pressure must be high enough to pierce the plasma and nuclear membranes, but not so high as to kill the cells. Microinject cells along the wound edge for a fixed period of time. With practice, several hundred cells can be injected during this interval. If injecting plasmid DNA, Incubate the microinjected cells at 37 degrees Celsius in a tissue culture incubator for 20 minutes prior to fixation. Then terminate transcription by treating the cells with 1 microgram per milliliter alpha-amanitin dissolved in growth media. This concentration effectively inhibits transcription for microinjected plasmids. Place the cells back in the incubator and allow the cells to export the newly synthesized mRNA. A single needle can be reused to inject multiple cover slips, although the needle needs to be replaced when the type of DNA or RNA that is microinjected is changed. Furthermore, once the needle is removed from the wand, it should be disposed of. To obtain an accurate measurement of the mRNA nuclear export kinetics, separate samples should be fixed at 0 minutes, 15 minutes, 30 minutes, 60 minutes, and 120 minutes post-alpha-amanitin treatment. At the appropriate time, aspirate the growth media and wash the cells twice in 2 milliliters of PBS solution. It is important to keep the cover slips moist by minimizing the time they are not submerged in liquid. Fix the cells by adding 2 milliliters of 4% paraformaldehyde in PBS. Incubate the cells for at least 15 minutes at room temperature. After washing the fixed samples 2 more times, permeabilize the cells with 2 milliliters of 0.1% Triton X100 in PBS. Again, incubate the cells for at least 15 minutes at room temperature. Finally, wash the samples an additional 2 times. To prepare the samples for hybridization, wash the cells twice with a wash buffer of 1x SSC solution containing 25 to 60% formamid. Then, prepare the staining chamber for hybridization by covering the bottom of a 150 mm petri dish with water and floating a piece of parafilm on top. Remove the water by turning the dish over, thus allowing the parafilm to adhere to the bottom of the dish. Manually remove any air bubbles. For each cover slip to be stained, pipette 100 microliter drops of the hybridization solution onto the parafilm. Note that the amount of formamid should be optimized for the particular fish probe being used. With the help of forceps, remove a cover slip with the fixed cells from the 30 mm petri dish. Using Whatman filter paper, dry the backside of the cover slip and wick excess buffer from the front side without drying the cells. Then, place the cover slip face down onto the fish solution. Repeat this process for each cover slip. After placing the staining chamber's lid back on, incubate the samples for 5 to 18 hours at 37 degrees Celsius. Prepare several washing chambers in the same manner as the staining chamber was prepared. For each cover slip, pipette 1 milliliter of wash buffer onto the parafilm of the washing chamber. Following incubation, remove the cover slips from the staining chamber by pipetting 1 milliliter of wash buffer next to the cover slip. The liquid should be drawn underneath each sample by capillary action. Then, remove the cover slips with forceps and place each onto a drop of wash buffer. Incubate at room temperature for 5 minutes. Wash each cover slip twice more for a total of 3 washes. For each cover slip, pipette 10 to 30 microliters of mounting solution with DAPI onto a previously washed slide. Each slide can accommodate 2 cover slips. Using forceps and Whatman filter paper, dry the back of the cover slips and wick excess liquid from the front side of the cover slip without drying the samples. Place each cover slip face down onto a drop of mounting solution. Mounted samples can be stored at 4 degrees Celsius. Here, the effects of alpha-amanitin on transcription of injected plasmid DNA is shown. NIH 3T3 fibroblast cells, which were pre-treated with varying concentrations of alpha-amanitin, were injected with T-FUTS delta I plasmid DNA and OG conjugated 70 kilodalton dextran. Each row corresponds to a single field of view imaged for OG 70 kilodalton dextran and T-FUTS delta I mRNA. It is evident that high concentrations of the drug completely inhibited production of T-FUTS delta I transcript, whereas this was not observed at lower concentrations. Shown here is a time course of mRNA nuclear export. COS7 cells were injected with T-FUTS delta I plasmid DNA and OG conjugated 70 kilodalton dextran. 30 minutes following, injected cells were treated with alpha-amanitin and incubated at 37 degrees Celsius for 0 minutes, 60 minutes, and 120 minutes. The cells were then fixed and stained with a probe against T-FUTS delta I mRNA and with antibodies against the ER marker TRAP-alpha. Each row corresponds to a single field of cells. For information on how to quantify mRNA export, please refer to the online text. Here, The 120-minute time point was blown up to demonstrate the co-localization of T-FUTS delta I mRNA in the ER. An overlay of the T-FUTS delta I mRNA in green and the trap alpha in red is shown. Information on how to co-stain mRNA with proteins can be found in the online text.
We've just shown you how to measure the nuclear export kinetics of mRNA by microinjection. When performing this procedure, it's important to remember that a successful microinjection requires proper preparation. Key steps such as DNA and RNA sample preparation, pulling needles, aligning optical equipment, and troubleshooting microinjection are described in our written protocol. When imaging and analyzing microinjected mRNA, it is important that quantification is limited to cells that received greater than 90% of the injected fluid into the nucleus. A successful nuclear injection can be assessed by visualizing the distribution of organ green dextran in the nucleus and cytoplasm. Please refer to our online protocol for more information about designing FISH probes and for using image analysis software for analyzing the distribution of mRNA in the cytoplasm and the nucleus. So that's it. Thank you for watching and good luck with your experiments.